I'm Duane Schulthess, and on this Vital Health podcast, I'm speaking to two Primrose project managers in charge of the project's data collection and management. Leiden Fagereng, project manager of Primrose, and the coordinator of the Precision Cancer Center at Oslo University Hospital. Liva, how are you doing?
I'm very well, thank you.
It's good to see you.
Thank you.
And Henk van der Pol, a Ph.D. candidate in the Leiden University Medical Center with a background in computer science and applied mathematics. Hello, Henk. How are you doing?
Hello, I'm doing great.
Primrose is structured around a drug Rediscovery Protocol, or DRUP trial. What is a DRUP trial? How does this differ from a normal clinical trial?
The DRUP trial protocol, which is based in the Netherlands, and the similar trials that you find in several European countries, separates from other clinical trials that is a more integrated part of the healthcare system. So it is scanning or screening all the patients that go through the molecular tumor boards in each country to include them into treatment arms in the trials.
So the population is an existing group of patients that you're drawing from a known asset, a known drug. Are these pre-approved drugs?
The drugs are approved for one indication, so this is off-label use in the trials, testing them outside their approved label.
So, Henk, from your perspective, you're drawing in a bunch of people to get tested that normally wouldn't be tested in new indications. What sort of a challenge does that create for you in trying to develop clinical trial protocols, manage the data collection? I mean, this sounds quite challenging.
Yeah, it is super challenging because you have such a heterogeneous population as well.
What do you mean by heterogeneity?
Oh, yeah, sorry.
No, that's fine. Go ahead.
The population is very diverse. There's a lot of variance between each patient because a patient is included in the trial connected to a treatment, a biomarker target, that is where the treatment is for, and the tumor type. So you can imagine that for a biomarker and different levels of tumor type, there are multiple combinations possible. So you have very rare cases where you, for the drug trials already existing for 8 years. So you, for the past 8 years, you've seen only 4 patients with a specific tumor type and biomarker target that could be connected to a treatment within the DRUP trial. So you have a lot of multiple cohorts with very few patients.
Right.
So you have like 300 cohorts with
4 or 5 patients.
4 or 5 patients. So it's really difficult to do some statistical analysis on it because you have only you have like 4 or 5 people to. So also the question is, okay, how do I merge my cohorts together? Maybe how do I make that statistically sense, or how is there even a rationale to merge biomarkers together or tumor types together to even have some statistical evaluation for it?
So essentially, Liva, you're looking at 300 orphan drug trials, essentially, on some level when you're kicking this off. That sounds hugely challenging. And as Henk has said, the numbers are low. How many sites are you working with to try and make this work?
As you said, these are national trials, so they are independent in the trials in each country. However, we merge the data between the trials. So given the total number of sites and hospital open in each country, we're talking hundreds.
Yeah. If a doctor has a patient that maybe is not responding to treatment, what would be the approach to get the patient involved in the trial? How does this actually happen?
So that's kind of the cue that since the patients go through the standard of care and their treatment lines, and also the molecular tumor boards are integrated as part of the standard of care, you already know from the molecular tumor board that they can be eligible for a treatment in these trials, so that they are already offered the treatment before they progress on their last line of treatment as part of standard of care.
Digging into that, so let's say you've got the patient in the tumor board. You know you're dealing with small numbers. You've got the biomarker validation. But your design is quite interesting. As Henk has alluded, you've got small numbers at the front end. So your protocol is you start with 8 patients in a cohort, and you validate that. Then you move to 16. So you're doubling each time. What's the rationale of having 8 to 16 to 30, cranking up 2 by 2 by 2 as it is?
So the number 8 and 16 is more of like, well, it's not random, but it's more the real design. It's a Simon 2-stage model.
Okay.
And in this 2-stage model, what you do first, you choose a true response rate, in this case 30%, to a negative response rate, which is less than 10% or 10%. And then you choose an alpha level, so that's in this case 7.8%, and then a power level, which is 85%. If you fill these numbers in, then the number 8 and 16 comes out for assignment 2-stage model. So that's where you get these numbers from.
So if you're using an alpha of 7.5, just, you're trying to infer statistical significance there. What would happen if you didn't go for 30? What if you went, say, okay, we're getting 20% response? Does that work?
Yeah, you could also do that, but this was decided 8 years ago. I think I was still in the first year of high school, but
well, for you, Henk.
Yeah. But what I know is that they chose at 30% to have really some significant benefit to see in each cohort. That's why they chose at 30%, I guess.
A balance between you need a scan and a certain number of patients to ensure that you pick up a potential significant effect. However, as we see, it's difficult to find that many patients. You can't kind of search too broadly, because if you say that you want 30 patients or 40 patients or 50 patients, you would never reach the numbers. So it's a pragmatic way to see how many patients do we find and how.
And that's why they also chose, usually they have an 80% power level, but now in this trial they have an 85% power level, because they want to have really some significant result in a cohort that actually
you're trying to crank it up a little bit more.
Yeah.
It's funny because we were very involved in adaptive licensing and adaptive approaches, and the big joke was, okay, you do a phase 2, you have 50 patients, 1/3 respond, 1/3 don't respond, 1/3, get sicker. And then you go to a phase 3, you get 500 patients, 1/3 respond, 1/3 don't respond.
Yeah.
So, I mean, it seems like that 30% is always kind of a magic number. I mean, Liva, you're in charge of optimization in Oncology Treatment Center. Do you see that? Does this hold through?
One of the really important aspects is, one thing is for the first kind of 2 stages, stage 1 and stage 2, but the really significant thing is also the re-evaluation to be more specific about the biomarkers for the stage 3 or the expansion cohort, so that you can learn something from the first stages. Because in the end, it's not about getting a good treatment to 1/3 of the patients you treat. You want to optimize so that you continuously are better to select patients to the best treatment alternatives.
Yeah, and if it also goes wrong for a really bad cohort, then you can also quit very early with 8 patients if you don't see any clinical benefit.
Yeah,
you have some decision making in the meantime already, but that's your strict sampling plan as well.
Sure, but these are approved drugs as well, so they should have a very well-known safety profile. Hopefully won't get too many surprises, I would hope.
Yes, but they still use it off-label, so there's not really there's still maybe some gray area in between.
There is, of course, the safety profile of approved drugs, but it's a difference between recently approved drugs. And we see that also for the pharma companies, that they will want a different degree of information about the safety level from our cohorts too.
You touched on something earlier. You're talking about a federated clinical trial design where you have 5 or 10 patients in one site, you got 5 or 10 patients in another site. Now, you're dealing with larger indications, I assume, so you're not limited in that sense, but how are you managing the data collection around multiple sites? That must be a big challenge. How do you deal with that?
Yeah, that is indeed a very big challenge. So the data collection within Primos is done via the TSD, and the TSD is a computer which is highly secured, based in Oslo, where all the cohorts that have been finished, so they have accrued 8 or 24 patients, they will be stored in that database.
So just to clarify, it's not really a federated, inside Europe, it's not federated data. They will all be collected into one server that's stored at Oslo University Hospital. So what we've done is that since there are separate trials, we identified a core set of endpoints that we share with the variables we share, and all pull out that from their ESRF system. So this is structured data from clinical trials that are transferred to the TSD or the sensitive server.
Your central server.
Yeah, our central server. And so the challenge is, of course, even though that it is structured data, it is predefined variables. However, you will still see the need for harmonization. For example, the definition of tumor types. What you define is a colorectal cancer. Do you include colorectal neck cancers? Do you don't? Do you use the ICD code? Is that sufficient? Does it these kind of details both on the tumor type specifically and also the biomarkers? Yeah. So it is a work on harmonization between the trials for these variables before we will be able to merge them, because complete response, for example, is quite well defined.
And also because you have federated trials, but still if you want to have some evaluation on a cohort to merge cohorts together, you still need to have some central computer at some point.
But you also need a uniform approach to coding, as Liva was saying. You can have two hospitals in the same city that have two EHR coding systems. They're using universal basis, they're using metric basis. You'll have different definitions of endpoint for progression-free survival, and this is all coded differently. They're using different ICD codes for the same stuff. How are you dealing with those problems? Because I'm sure you have a shorthand in Oslo that you work with, but other hospitals probably have a very different approach.
For some of the variables is quite easy and structured. So the difficult part is, of course, how to add procedures for the data merging that you ensure that you compare apples and apples in reality.
Yeah.
And to a certain degree, you also need to accept that there is variations and you need to be aware of the variation and just state it.
What about doctor notes? A lot of the stuff you need is actually in medical notes at the bottom of the chart.
Yeah, but this is not really that challenging for clinical trials because they're all adding the information into a structured format in the electronic case report form. So that's a digital tool that they use to add. So we're not dealing with the same trouble as real-world data using directly data from the hospital systems, because that's more chaotic. Here it's more organized, that you, for example, have a tumor types, you will have a pull-down menu that you choose from, so you don't need to interpret the doctor notes, which is, of course, making it easier.
Boy, oh boy, that's hard. Europe has been spending a lot of time on things like OMOP, observational data platforms and things like that. Henk, do you see that as a potential way to solve a lot of this trouble?
Yeah, definitely. We're definitely going to implement as well the OMOP model because, well, as you say, you have different ECRF system, and not that the ECRF system is between hospitals, but it's between countries, so it's even more diverse as well. And the OMOP model really allows us to connect each variable, standardize it to an OMOP variable, to an OMOP code, and then collect everything and merge everything into one list, so to say, for all the variables we have, all the variables we collect as well.
Do you see OMOP as becoming a de facto standard? You have Cerner, you've got Epic, you have all these competing platforms. What do you see happening? Because you're on the knife edge of this. You're the person who has to deal with this stuff.
Yes, yeah.
I mean, what do you see happening?
As far as I know, the OMOP model is the only one that really is implemented to deal with this ECRF's problem, to deal with this clinical data, with outcome clinical data. And it has also a really big community, forums every year, conferences, and really a big forum online as well to really harmonize and standardize new variables as well. Because, for example, biomarkers, there are, I don't know how it goes with biomarkers, but probably every year there will be a biomarker really well defined or newly defined as well.
Yeah.
So I think for that you need a really good community that really updates every variable and standardizes the variables.
You can say for our use, we could have chosen any standard. We don't need an established standard to merge our data internally in the DRUP-like clinical trials. However, by standardizing them to an established model, we will also enable the possibility to do federated analysis with trials or outside of Europe, because we are governed by the same GDPR legal framework. But if we want to merge with the South Koreans or the Japan or the Tapur study in US, they could, in principle, use our OMOP model and do the same standardization of their variables, because they have the exact same variables in their trial as we do. So they could standardize towards the OMOP model, and then we can do a federated analysis with our data and their data, because they also have the same drugs and the same cohorts and the same biomarkers. Again, moving to a more global level.
Obviously, you can deal with the manual indications when you're dealing with 8 or 16 patients in your early stage stuff. But if you start getting 500, 1,000, 640, this starts to become really onerous, because the complexity doesn't go linear, it's exponential, right?
Yeah.
So you end up pulling your hair out. It ends up being a lot of work. You did mention GDPR. Obviously, there's been a lot of hemming and hawing around GDPR. Do you see that as an impediment to what you're trying to do?
Again, since this is clinical trials, the patients signed the informed consent form. So it makes it easier, both that we are all, of course, governed by the same legal framework, but also that they sign informed consent to share the data. So in reality, that's really not been the trouble for us.
But if you wanted to say prospectively go through the clinical trial register of the hospital and say, do we have patients that fall into this criteria? We have patients who are non-responders.
Here, I think it's different practices in different countries, but usually for the molecular tumor boards, they will know what's in the pipeline for coming drugs and also reach out. So for example, in Norway, we have an informed consent prior to entering the molecular tumor boards, prior to the precision diagnostics, again enabling us to keep a register, to keep an overview of the patients, knowing that if you get your tumor sequenced and you get a discussion in molecular tumor boards, if a new drug or a new trial enters Norway that you would be eligible to, they will scan through the last year's patients to see if there is anyone eligible so that they can contact and get access to the treatment, to ensure that it's not just a one-time discussion, it's something that's kept in the patient journal.
When you're dealing with the data that you have, everyone's in a tumor board here in the Netherlands that's already been opted in.
Yeah, there is a molecular tumor board as well in the Netherlands. It works quite similar as in Norway as well.
Obviously, we're talking about federated data. This has come up a couple times. 80% of all oncology indications have generally been released with an orphan indication. This is historically how it's gone. Do you see a challenge of trying to work with federated data in orphan indications? 80% of available orphan indications in Europe are less than 1 in 1 million, so you're literally talking a maximum population in Europe of 500 people.
Yeah.
Now, the large orphan indications are actually 1 in 10,000, roughly. So they're the large orphan indications, and that's where most people develop.
Yeah,
because economically, because if you're only targeting 500 people and you're spending $600 million to $1.1 billion on development, it gets a little expensive.
No, very clear, yeah.
A lot of the cancer indications are launched as orphans. You're dealing with federated data, as we've discussed. That can be a challenge. How do you square that circle?
Yeah, so again, this is kind of the main drive force for doing this federated analysis and merging the data and do the collaboration between the trials is to increase the inclusion area. In reality, from 5 million in Norway to 70 million if you go through, and again expanding to, if you include now Germany and Spain, you're really getting a large population you collect data from.
Mm-hmm.
So in that case, you get maybe 1 patient in Norway, 2 patients in the Netherlands. But if you collect all these federated data sets together, at some point you can complete a cohort or fill cohort.
That works because you're centralizing the data.
Not necessarily because we're centralizing the data, because we merged the data. We could have done the federated analysis with fewer patients. But again, since we are all a part of Europe, we can do it. It's easier to do it.
One of the other challenges around cancer, particularly with a lot of the diagnosis and response, is imaging. From the standpoint of biopsies and pathology, imaging is really, really important. How are you dealing with imaging in the context of the project?
So again, luckily, these are part of trials, so it's a very standardized tumor assessment during the RECIST and eRECIST or RANO, depending on which tumor type you're having. So it's a very standardized way to do the tumor assessment.
So do you get the images transferred in, or are you just getting the results?
No, you're just getting the results.
Yeah, we only get the results.
Yep.
Now, Henk, there's been a lot of work on AI for imaging analysis, and a lot of the data, there were several publications in the New England Journal of Medicine last year that showed that AI models are actually getting quite good around imaging analysis. I mean, how are you seeing this evolve here in Leiden?
Well, this is more of like a two-way question. Is first that, these machine learning methods or AI imaging methods, they work really good in AI because you have a high dimensional data set and you want to compress it into sort of a yes or no question, right? If there's a tumor or not. So that's where these machine learning methods work really good. And what we're working more is we work with the outcome, so more with the clinical data and see, if there's any clinical benefit, yes or no, really. So we have a less high dimensional data set. So for us, the more standardized statistical methods work maybe better. Also because we have not like AI imaging methods, we don't have tens of millions of data. We only have we have very few data.
Again, which is a help, really.
Which is a help, yeah. Which is actually kind of positive, yeah. Beside this project, there is a lot of work in AI imaging, of course, in the Netherlands, especially I think also in Leiden, yeah.
Yeah, because that's been a huge constraint.
Yeah, it's true.
Yeah.
I mean, that's, pathology on imaging has been a huge, huge constraint. And it's been more art than science.
Your words, not mine.
Yeah, I'm sorry. With all due respect to all pathologists out there, please. No, it does seem to be an area that's quite evolving. I mean, how are you seeing this in Norway as far as using machine learning applications for imaging?
It is a long way from a proof of concept to implementation into healthcare. But again, I guess as all European healthcare system experience, and especially maybe us up in the north, that we have a constraint in the human resources.
Yeah.
So we need the kind of pushing the digitalization forward is the lack of radiologists and pathologists to do the assessments.
As part of Primrose, one of the aspects that you're trying to incorporate are quality of life. These have been quite a lot of discussions the last time we were together. How are you bringing that to the project?
Some of the trials already have quality of life already implemented, and some trials are now implementing quality of life forms. So they will be shared variables as the same as the clinical variables. They will also be stored in a common data server.
How do you deal with quality of life metrics? How do you see that? Is it just another tool in the toolbox? How do you apply that to your analysis?
The quality of life data really gives a new perspective on your clinical data as well, because it gives a more perspective of how a patient is actually feeling or actually doing during a treatment and trial. And
so because we made a statistical analysis plan for the Primrose dataset with the Primrose endpoints and quality of life is part of that.
Yeah.
These endpoints in the analysis plan. So they will be incorporated and reported on. So the aim is, of course, to when we either close a cohort at 8 patients due to no benefit or 24 patients because we reached stage 2, is to publish and report on this, including the quality of life data.
And from a practical standpoint, 10, 15, 20 years ago, there was some pushback against these things into analysis in clinical trials. Are you seeing more acceptance?
If you say 10, 20 years ago, this immunotherapy cohorts, this trial, it couldn't exist right now. That there is even space allowed for quality of life surveys really suggests that there's more room and there's really some improvement in the past 10 to 20 years, to have these quality of life.
And we see quite a clear acceptance for having quality of life, but a varying degree, depending on who you talk to and which professions. Some would love the EQ5D, somebody wants the QLQ30. I mean, there is, depending on how you use it. And
yeah, I mean, everyone has a certain flavor and a certain desire, but it is interesting that it's seen now as a vital part of the pathway. I mean, you're incorporating it, and there's a reason why.
Yeah, and then because there is, of course, that we have a strategy not just for the results from the stage 2 or kind of the 24 patients, is the strategy for how to move a drug all the way into permanent reimbursement and implementation into the healthcare system. And during that pathway, it is, of course, a lot of the decision makers that rely on the quality of life data in order to make their decision.
And of course, a lot of the patients are increasingly demanding that as well.
Absolutely. And I think for the long-term aim for the quality of life is to now it's a well-established quality of life form that mainly will be used for reimbursement decision later, but it is an aim to also have a more interactive quality of life that you can act upon as part of the clinical decision so that the doctors and the patients also can use it in their assessment of the treatment.
So what are the next steps for the project? What's in store for the next 6 months? Where does Primrose go?
So a part of the merging of the data is really implementing this OMOP model, this OMOP framework, to really standardize and harmonize, standardize first, this is a better term, I guess, is to standardize the first cohorts that are actually actively being merged. So that's actually that's the main point, really, for the next half year, I guess.
At least for you.
At least for me. That will keep me up at night, I guess.
And during the day, and
during the day,
some of the weekends
24/7.
Yes.
Yeah, so it's because that's a very important part to get the data sharing platform and the optimization and get this kind of rolling in there. Because we have monthly meeting where we go through the new cohorts and see where we included and kind of where we're going to move forward, which cohorts that have been filled and which will be closed or open for a stage 2. So that's a super important part. And then there's, of course,
also to create an external control arm, because here in the drug trials you only have a treatment arm. So you also want to mimic a randomized clinical trial by creating an external control arm by looking at the available real-world data of genomic data and see if we can create a control arm to evaluate these treatment arms within the DRUP trial.
So are virtual controls being more accepted now? I mean, 10 years ago when we started doing this work, there was quite a bit of pushback. How is this being adapted now?
I think there's still a lot of pushback because there's a lot of skepticism because your trial is not a randomized clinical trial. So at the start
one could debate that.
Yeah, of course.
One could debate that.
It also really depends on your real world data, right?
Sure.
It depends on the quality of data you can use because you can give an argument. Well, okay, these patients are in your trial because they have such very strict inclusion criteria, but it might not hold for your real world data. So that's also really a big, well, not a problem, but it's also really something you need to describe and really be aware of. That it's not something you can just plug in some data and see, okay, I have some better overall survival rate because it is in my real world data. So you need to really think really thorough.
One of the main challenges we saw was you can't go too far back because clinical practice is always evolving, particularly when you're dealing with oncology. If you go back before the introduction of monoclonal antibodies, that has a huge impact on your survivability. So there are certain things you have to be very conscious of when you're doing a virtual control. We find that we're doing a lot of this in comparative effectiveness, and it can work, but you're right. You got to be very careful.
Yeah, you have to be very, very careful.
Liva, let's say practitioners, other people like you running hospital efficiency, dealing with data, they want to get involved with Primrose. How do they do it?
Well, the easiest one is to go into our webpage, send in a contact request. So primrose.eu.
Primrose.eu. Is that one word or a hyphen in the middle?
It's a hyphen in the middle.
Prime-rose.eu. Fantastic. Henk Vanderpoel, thank you very much.
Thank you. Nice to meet you.
Liva Fagereng, thank you very much, Liva. It's always lovely to see you.
Thank you very much. Thank you.
And thank you for your time. The executive producer of the Vital Health podcast is Duane Schulthess. Our editor is Mark Brodeen. Our project manager is Gwen O'Loughlin. The Vital Health podcast is a production of Vital Transformation LLC, copyright 2024.